my PhD was part of this MBIE project, which is great, working alongside Manaaki Whenua, Plant and Food, Scion, Ag Research and UC, while studying at Lincoln. So my PhD was titled Reduction of Nitrogen Losses by Manipulating Carbon Inputs in Pasture Composition. And so now I work at Ravensdown with the Innovation and Strategy team as a scientific officer. So why is nitrogen so important? Nitrogen, as we know, is essential for plant growth. However, there are some issues with it. Nitrate can be lost into the waterways, causing eutrophication. And nitrogen can also be lost as nitrous oxide, a potent greenhouse gas and an ozone-depleting substance. So we want to keep that nitrogen in the soil for farmers, for plant growth, and reduce those environmental impacts. So we want to study ways of how we can reduce those losses. So we conducted lysimeter experiments. So all our experiments were in this stony Balmoral soil, which are a joy to dig lysimeters into. So once we've dug them in, we take them out, we process them, and we put them into lysimeter trenches here. Then we apply urine to them because that's the hot spot for nitrogen losses. We then collect gas samples, leachate samples, and herbage samples. So every lysimeter also has a corresponding soil block which receives identical treatments to the lysimeters. So this allows us to take destructive soil samples without interfering with the drainage and the pasture and the lysimeters. So it gives us an insight into what's happening to the soil. So from this, we measured from our gas samples, we could quantify nitrous oxide emissions. From herbage samples, we could quantify the total yield, plant nitrogen uptake. And from our leachate samples, we could quantify nitrogen leaching, ammonium leaching, and total drainage volume. From our soil samples, from our soil blocks, we could get soil mineral nitrogen, nitrate, ammonium, soil microbial nitrogen, and the ammonia oxidizing bacteria gene abundance. So these bacteria are really important. These are the bacteria that convert ammonium, NH4+, which is held well in soil, to nitrate, which is negatively charged and can be leached easily. So these bacteria are really important for and are responsible for nitrification. So that's why we looked at them. And so we sampled on days 0, 1, 7, 14, 28, 56, and 112 days after urine application. So we could see what the effect urine had and see what's going on in the soil over a long period of time. So that's a summary of what we measured and how we measured it. So what do we look at? What methods do we look at ways of reducing nitrogen losses from farms? So we looked at different urine nitrogen rates. We also looked at different urine carbon compositions. So we collected urine from cows grazing different diets. We standardized the nitrogen rates to quantify what effect that urine carbon has on nitrogen losses. We also looked at different crop types, different pasture species. We also looked at for biological nitrification inhibitors. So biological inhibitors are those ammonia oxidizing bacteria. And we also looked at applying artificial carbon inputs to cause immobilization and reduce nitrogen losses. There's also some other ways of reducing nitrogen losses that we didn't look at, but quite interesting. Standoff pads, catch crops, nitrification inhibitors, and irrigation, having efficient irrigation to reduce that drainage volume. So in the first year, we looked at different crop and urine types. So we published a paper on nutrient cycling in agroecosystems. So we looked at cattle diet and winter plant growth effects on nitrogen losses. So we had two crops. We had fodder beet soil, then we pulled the fodder beet out with simulated grazing as would be on the farm. So it was bare fallow soil, which then had urine applied. We also had pasture, which had urine applied as well. So we applied urine at 700 kgs of nitrogen a hectare, like a typical cattle urine patch. We also collected urine from cows on 2 different diets. So we collected urine from cows grazing fodder beet and cows grazing ryegrass white clover. So the fodder beet urine obviously had lower nitrogen concentrations in it, so we added urea to standardise them all up to 700 kgs in a hectare. So we could look at the effect diet and urine carbon composition had on nitrogen losses. And so there's 5 lysimeters, replicates for each. So we saw that crop type and cattle diets affect nitrogen losses. So we saw the perennial ryegrass white clover pasture leached 77 to 93% less than the bare fodder beet soil. So winter plant growth is really important for reducing nitrogen losses. We also really interestingly saw the fodder beet urine, even at the same nitrogen rate, when applied to the pasture, leached 64% less nitrate than the ryegrass white clover urine. So we were really interested and we thought, why is this happening? This is statistically significant. 64% is a big reduction. So we looked at the soil data for why that fodder beet urine, even at the same nitrogen rate, is leaching less than the ryegrass white clover urine. So this is the ammonia oxidizing bacteria gene abundance. And so we can see the fodder beet urine over the experiment. So this is the gene abundance on the y-axis and the days after urine application on the x-axis. The fodder beet urine was significantly lower for AOB gene abundance. So we saw 36% lower AOB population and subsequently 31% lower soil nitrate. So something in that fodder beet urine is causing a biological nitrification inhibitory effect, which we thought was really interesting. So to reduce nitrogen losses, we saw that fodder beet urine leached 64% less than ryegrass white clover urine applied at the same nitrogen rate. So cattle diet affects nitrogen losses. In the first year, we also published another paper, effects of adding readily available carbon to soil on nitrogen losses. So we looked at 2 different crop types, perennial ryegrass white clover pasture again, and lucerne. So we applied urine again at 700 kgs in a hectare. And we also applied carbon inputs. So we applied carbon at 12 tonnes of sucrose a hectare and 24 tonnes of sucrose a hectare for the ryegrass white clover. And lucerne we had controls as well that received no carbon input. And in lucerne there's another treatment with 12 tonnes of sucrose a hectare. So we applied sucrose just before we applied the urine to stimulate microbial growth. And so we saw carbon addition and crop type affected nitrogen leaching losses. So the application of carbon reduced nitrate leaching under perennial ryegrass white clover and lucerne. So under ryegrass white clover we saw 75% reduction in nitrogen leaching when we applied 12 tonnes of sucrose per hectare, and 91 for the 24 tonnes of sucrose hectare treatment. And the lucerne we saw a 51% reduction when we applied 12 tonnes a hectare of sucrose. And we also once again saw the more winter active perennial ryegrass white clover pasture leached 67% less nitrogen than the winter dormant lucerne. So just by going from lucerne to perennial ryegrass white clover, a more winter active crop, you can reduce nitrogen losses by a lot. So we also saw that adding this readily available carbon didn't actually increase nitrous oxide emissions significantly, which we thought it might have. So why was the leaching lower under these carbon inputs? We saw that, so here's the soil mineral nitrogen on the y-axis, and on the x-axis is time, days after urine application, and on this one is the ryegrass white clover. So there's the control treatment, 12 tonnes and 24 tonnes of sucrose over the varying days. So you can see over the experiment, the soil mineral N was much lower under the carbon treatments, unsurprisingly. Same again with the lucerne. So here's some data from Brendon and his team. So we looked at the microbial biomass N, and so nitrogen's not been leached, it's not in the mineral N, where is it? So the microbial biomass N is significantly higher under the carbon treatments. So that's carbon is stimulating microbial growth, which is sucking up more nitrogen, reducing soil mineral N and subsequently nitrogen losses. So from that first year, the crop type, we saw really significantly affected nitrogen losses. So to reduce nitrogen losses, winter plant growth is extremely important. The bare fallow fodder beet soil leached a lot, even going to a winter inactive lucerne, reduced leaching by 10%, but going to that perennial ryegrass white clover pasture with some winter growth, reduced nitrogen losses by 2/3. So winter plant growth is extremely important for reducing nitrogen losses. So in the second year, we looked at different pasture types. We looked at perennial ryegrass white clover, and plantain. We had some issues this year with plantain persistence and piranha damaging the plantain, so the results weren't a fair comparison. We also looked at urine from cows grazing ryegrass white clover and plantain. And so we saw no significant effect of having plantain in the urine. We also looked at urine rates, so 500 kgs of nitrogen hectare versus 700, and this is where we got some really good results this year. So we saw that lowering the urine nitrogen rate 28% resulted in a 39% reduction in nitrogen leaching. So we got really good bang for our buck. If you can reduce your urine rate 28%, we saw a 39% reduction in nitrogen leaching losses. We also saw the same for nitrous oxide emissions. Lowering that urine rate just 28% resulted in a 38% reduction in nitrous oxide emissions. So it's a really efficient way of reducing those nitrogen losses, is lowering nitrogen concentration in urine and or reducing cattle nitrogen intake. So for the third year experiment, we looked at the timing cattle urine had on nitrogen losses and pasture composition had on nitrogen losses. So we had 3 pasture types: traditional perennial ryegrass white clover, perennial ryegrass white clover and plantain, and Italian ryegrass white clover and plantain. And we applied urine in February, March, April or May. We applied it at 700 kgs nitrogen a hectare. So we saw the nitrogen leaching losses were actually surprisingly high. In these warmer months, we thought there'd be more plant uptake and lower losses, but there was quite high losses in February, March and April, which is interesting. And we also saw that incorporating the winter active Italian ryegrass and plantain reduced nitrogen losses by 24%, and adding plantain into the mix reduced nitrogen losses by 14%. So having that winter active Italian ryegrass and plantain added into pastures can reduce nitrogen leaching losses. So why were the losses lower under the Italian ryegrass and plantain? We attributed that to the mid-autumn to mid-winter plant growth. So once again, plant growth is really important. We saw perennial ryegrass growing 1.8 tonnes of dry matter over that period, with plantain went up to 1.9, but the Italian ryegrass, white clover and plantain went up to 2.3 tonnes of dry matter a hectare over that period. So that increased plant growth from the Italian over that winter period was really important for reducing nitrogen losses. So the main takeaway points from this: to reduce nitrogen losses, incorporating Italian ryegrass and plantain into pastures can reduce nitrogen losses. Reducing urine nitrogen concentration, so having low nitrogen feeds and supplements, or spreading the amount the urine patches over a greater area, diuretic effect, can be a really effective way of reducing nitrogen losses from grazing systems. Winter active crops reduce nitrogen losses. And interestingly, we saw phosphate urine has a potential biological nitrification inhibitory effect and can reduce nitrogen losses. We also saw that applying readily available carbon to soil can significantly reduce nitrogen leaching losses without significantly increasing nitrous oxide emissions. So I'd like to thank the funding from MBIE for the Reducing Nitrogen Losses from Farms program and the partnership between Manaaki Whenua, Lincoln and Plant and Food and Scion. So thank you guys. So the question was, in the last experiment, did we measure nitrous oxide emissions? We did, but there was no significant effect in March, April, May, and I think Italian may have significantly reduced nitrous oxide emissions in the February urine treatment, so it wasn't too significant. That's why we haven't put in the presentation. Good question. So the question was about the urine nitrogen concentrations and the urine carbon concentrations in that first year study to do with the phosphate. So the urine nitrogen concentration, initially when we got the urine from the cows, the urine nitrogen was lower under the phosphate, but we added urea to standardise them both up to 700. So the urine nitrogen concentrations, it was 2 litres of about 6.9 milligrams of nitrogen a litre. So it was the exact same nitrogen rate between the two treatments. We also did look at the urine carbon composition. So that's initially we thought, phosphate's a really high carbon crop, quite low nitrogen. We thought maybe there's more carbon coming out in the urine eaten by the phosphate, and that might be causing immobilisation like we saw with the sugar. So the urine carbon concentration was higher in the phosphate urine, but we didn't see an immobilisation effect. The amount of carbon was insignificant really. So it wasn't a mobilisation effect. The total soil mineral nitrogen was the same, which really surprised us. And then we looked at the makeup of the soil mineral nitrogen, and we saw the phosphate urine had much higher ammonium and lower nitrate than the ryegrass white clover urine. So we saw it wasn't an immobilisation effect. It had lower soil nitrate, higher soil ammonium, suggesting a nitrification inhibitory effect. And then we got the DNA data as well from the ammonia oxidizing bacteria, and that population was, as that slide showed, significantly inhibited. So that's why we've attributed that to a biological nitrification inhibitory effect. There's also another paper that found lower nitrous oxide emissions from phosphate urine versus kale urine. So this is the only paper that's found this interesting detail. Yeah. So, yeah, I think it'd be really interesting someone to further study why that phosphate urine is having that effect.
It does sort of, beg the question perhaps of you, could you speculate on what might be in that urine? I mean, or is there any literature work to suggest what
There's no literature work on the secret ingredient in phosphate urine, but I think it's definitely worthwhile someone looking at what carbon source, I assume, is having that nitrification inhibitory effect in that phosphate urine. So the question from David was, do these findings apply to other soil types? So I think a lot of them generally would. I think winter plant growth would be really important for every soil. I think those urine nitrogen concentrations, the effect might be even bigger on those larger soils that can have more of a buffering capacity. So I think it's important to study these nitrogen losses on all the different soils, but I think most of these things would apply to our soils, but stony soils do have their unique challenges, and I wouldn't want to extrapolate too far, but I assume most of these things, the rules would, the general takeaways would apply for most soils, but every soil is unique, so it's hard to generalise too much. Yeah, sorry, Paul's question was, adding the carbon resulted in increased immobilisation, more nitrogen held in the soil microbial N. Once those microbes die, do you think it will increase leaching? So we did that experiment for a whole year, so we looked at drainage for a whole year. And so I would say that a lot of that nitrogen that the microbes have that's sucked up over winter, well, they would slowly die off over summer, and that nitrogen would be sort of, we apply nitrogen fertiliser through summer because we've got all that plant growth to take it up, and there's very little losses then. So I think just by shifting that sort of time period sort of more to summer, instead of having that mineral then available, then to have that mineral available becoming slowly available over the season would reduce losses a lot. I don't think it would be, kicking the can down the road at all. I think, like we looked at, the drainage concentrations over that whole year, and they're significantly lower the whole time. There's no big spike at the end when the microbes died. So I'd say no.
Just wonder if you could just comment a little bit more on that sort of premium you get with the lower end addition rate, with that sort of bump you get in terms of 28% lower and then almost 40% reduction in both directions. Do you want to comment a little bit on?
Well, I guess the soil has a bit of a buffering capacity. You can apply a certain amount that the soil can hold and the plants can hold, and what's excess to that will be lost. So if you're, you apply, say the soil can hold 400 kgs and 500 goes on, you're only losing 100. If you got 400 buffering and 700 is applied, you're losing 300 sort of thing. So it's that's a really basic way of summarising it. But I think that's the reason is soil has a bit of a buffering capacity, and reducing that nitrogen losses down to close to that buffering capacity is why we saw that bang for the buck with reducing urine nitrogen concentrations, resulting in much lower nitrogen leaching and nitrous oxide emission losses.